So our third presentation, our final presentation of this particular session is from Fergus Imrie, who's a postdoc who joined our lab this year, and it's on self-supervised learning for genomics. So Fergus, whenever you're ready, please go ahead and share your screen.
So thanks for that introduction, Nick. And as Nick mentioned, my name is Fergus Imrie and I'm a postdoc in the Schaar lab, actually based at the University of California, Los Angeles. And as Nick mentioned, I'm going to speak about self-supervised learning for genomics. And so while the application focus of what I'm going to present today is on genomics, I do want to say that the methods I'm going to present here can indeed be applied to other data. Also, as Mihaela mentioned at the beginning, typically linear models have been applied in genomics, and the two methods I'm going to present today are our lab's kind of first steps towards really extending this to a much broader, different class of models that are able to capture complex nonlinearities in genomics data. So first off, I'm going to discuss self-supervised learning, what it is, and why perhaps we need it. So the central motivation behind self-supervised learning is that in many circumstances, we don't have sufficient labeled data. And actually obtaining additional samples would either be very costly or in many cases not possible at all. So if we consider the supervised learning problem shown here on the slide, if we want to fit a model to this labeled data set, we should be able to do so given access to sufficient labeled data. However, in many cases, this isn't actually available, as I mentioned, and our ability to learn a performant model can decrease considerably, primarily due to overfitting the limited quantity of labeled data we have. And this scenario is very common across many scenarios in healthcare, genomics, and other omics situations. And therefore, using unlabeled data is critical. Thankfully, in many cases, we do actually have access to large quantities of unlabeled data. And this is where self-supervised learning can come into play. So self-supervised learning adopts a two-stage training process. In the first step, we pre-train a model using unlabeled data. And then using a small quantity of labeled data, we transfer the insights learned on the unlabeled data to this labeled data set and fine-tune the model using this limited quantity of labeled data. Now, we're obviously not the first ones to propose self-supervised learning, and indeed in other domains such as computer vision, for example, image classification, or indeed natural language processing, self-supervision has proved hugely successful already. However, in genomics or even medicine more generally, we're not concerned primarily with these data modalities. And indeed, we're concerned with tabular data that is unique and actually fundamentally different from those types of data. And as a result, the methods proposed for, say, image recognition aren't readily applicable since they primarily rely on the inherent structure of images or language themselves. In contrast, tabular data has no explicit structure, and any structure we need to actually learn by the methods we're proposing. So the first method I'm going to discuss today is a method called VIME, developed in the lab about a year or so ago by Jin-Sun Yoon and collaborators, that introduces a novel self-supervised approach for tabular data. So I'm going to briefly explain how VIME learns from unlabeled data. And to do so, I want you to consider the left-hand side of the figure shown on the slide. So we first take an unlabeled data point and then corrupt this sample by masking some of the entries at random and replacing them with other values drawn from the marginal distributions of those features. We then train an encoder model to predict which values were corrupted and what their original values were. Now, in order to do this, the encoder must learn how the features are related to each other, and thus must learn the inherent structure of the data to perform these tasks, which will then make learning on the labeled data easier. As a brief illustrative application, VIME was applied to genome-wide polygenic scoring. So unfortunately, I don't have time to go into details, but for all 6 of the blood cell traits considered, VIME performed all of the benchmark methods tested, really demonstrating the power and the ability of self-supervised learning to outperform linear models for these complex data sets. Now, in the remainder of this talk, I want to go beyond standard prediction tasks and move towards discovery. And so what do I really mean by a standard prediction task here? So in standard prediction tasks, we have features, and in the case of genomics or omics, quite a large number of features, and we wish to predict some outcome. So while this kind of standard setup can achieve high performance, it doesn't inherently provide any scientific insights. So why was such a prediction made? Which feature resulted in the outcome of interest, such as an observed phenotype? Which in many scenarios isn't particularly helpful. Now, one solution which Jonathan touched on earlier is these post-hoc interpretability methods. What happens if we can do something quite different? And instead, we could remove the majority of the features and actually predict only using a small number of highly relevant features. And this is, of course, feature selection, which is not a new field by any stretch, but results in a much greater understanding of the outcome of interest and can even reduce cost in many scenarios, since you only have to measure these small number of variables. And finally, often this can improve generalizability of the models, since there's fewer features with which to overfit to, especially in the limited data scenarios. However, there's two significant challenges that I want to discuss when we want to come to perform feature selection. The first mirrors the challenge we had before, and that is of limited labeled data. Without sufficient data, feature selection is likely to pick up on spurious relationships between the feature and the label and not actually identify the important features. The second is of intra-feature correlation, which can confound feature selection by choosing a variable that isn't actually responsible for the outcome. For example, if we consider the data generating process shown in the middle of the slide, let's say we have a gene X1 that when highly expressed causes some phenotype in the population Y. But actually by being highly expressed, gene X1 also causes gene X2 to be highly expressed. Obviously in this scenario, the relevant feature that we wish to identify is feature X1, but feature X2 makes this very challenging since this gene is highly expressed whenever gene X1 is highly expressed. So I'll now describe our recent work called CEFS that aims to solve these two challenges for feature selection. First, to address the problem of limited labeled data, we adopt a two-stage self-supervised training procedure similar to the approaches I've described thus far. In the first phase, we fix the selection probabilities of the subset generator that selects features and train an encoder network using similar objectives to the approach of VINE that I've described before. Then in the second phase, which uses labeled data, we fix this encoder network and jointly learn a predictive network together with selection probabilities that determine which features are selected, all while trying to minimize the number of selected features. A key component of our method is actually how the features are selected. And this enables us to tackle the problem of intra-feature correlation between variables that might confound feature selection. And to do this, we explicitly model potential correlations between features via the use of a multivariate masking or selection process. And so instead of selecting features independently at random, we select features using a multivariate Bernoulli distribution that selects highly correlated features together. So this has a couple of benefits. The first of which is in the self-supervision phase. This prevents the model from trivially predicting the values of other masked features using highly correlated features, since the features will either be jointly masked or jointly not corrupted by the masking process. In the feature selection process, by jointly selecting features that are highly correlated, it forces the model to really decide which feature is most predictive of the target outcome, therefore not selecting multiple highly correlated features when only a small number of them are likely directly related to the outcome. To demonstrate our approach, I want to briefly describe an example on a toy dataset. So consider a dataset where only one feature is relevant for the target outcome, but there are 9 irrelevant, highly correlated features. On the left-hand side, using our proposed gating procedure, which uses correlated gating vectors, you can see that the model quickly learns to identify the relevant feature, as well as assigning very low selection probability to all of the irrelevant features. In contrast, on the right-hand side, using independent gating vectors, while the model correctly picks up the relevant feature, it also assigns high selection probabilities to several of the irrelevant features, demonstrating the benefit of our approach. I'll finish with some further results from our method CEFS on a number of datasets, including a transcriptomics dataset and a proteomics dataset, where we validated our approach both quantitatively on the left-hand side and shown qualitatively on the right-hand side. And our results here show that the features selected by Cephs are indeed highly predictive of the target outcome, and also that many of them can be validated in the scientific literature. If you'd like to know more about either of the works I presented, please see our papers or feel free to ask any questions in the Q&A if you have them. And thanks very much for your time. I hope you enjoy the rest of today.